Hi, I'm David Hafler, and today I'm going to give a brief overview of what we believe causes multiple sclerosis in relationship to emerging therapies. In terms of exactly what is multiple sclerosis, we know from over 100 years ago that the fundamental pathology of MS is in the white matter. One sees on autopsy lesions in the white matter, be it around the ventricles or in the brainstem. If one looks under the microscope, takes one of these lesions, put them under the microscope, one sees inflammation. The inflammation consists of both B cells, plasma cells, and both T cells of the CD4 and CD8 lineage. And we'll get back to B cells at the end as one of the emerging therapies are monoclonal antibodies against B cells. MS is a genetically mediated disease with now over 100 allelic variants, each with a very small effect size, have been identified that lead to the autoimmune disease in relationship to unknown environmental factors. So what do we know about multiple sclerosis? And this is really a brief summary of what I will talk about. One, if it's an autoimmune disease, that means the T cells must be directed against some autoantigen. The question is, what is the antigen? And the answer to this, as I'll show you, is there are multiple target antigens that the T cells are directed against. There are defects in immune regulation. We all have autoreactive T cells in our circulation, and a number of these immune defects have been identified. But there's not just one immune defect. There are multiple immune defects in terms of a population of T cells we'll discuss called Tregs and TR1 cells. And then what is a genetic architecture? As I said, multiple sclerosis is a complex genetic disease with multiple common genetic variants causing the disease. And what we've discovered is that these allelic variants, which cause MS, are predominantly in immune genes. In fact, are shared among different autoimmune diseases. And finally, what is the environmental trigger? And as I'll discuss, there are multiple environmental triggers for the disease, which may eventually lead to other novel forms of emerging therapies. So the first we'll discuss what are the target antigens for this autoimmune disease. Give first a very brief overview of basically the field of immunology. What we know is that a cell called a macrophage are antigen-presenting cells which present antigen to the T cell and to the T cell receptor. The T cell receptor recognizes the antigen MHC complex. Dr. Riley will be discussing one of the present therapies for multiple sclerosis, which is glutathione acetate or Copaxone, which is a molecule that binds the MHC and interacts with the T cell receptor. And here's a cartoon, actually a real histogram of a T cell receptor identified from a patient with multiple sclerosis. You see it recognizing a peptide in the groove of the MHC molecule. And this is really a central tenet to the field of immunology, the fact that one has a T cell receptor which can recognize an antigen in the context of this molecule called MHC. Our laboratory and many others over the years have identified different target antigens and we've used these assays to measure the frequency of autoreactive T cells. By doing these types of assays, we've identified particular epitopes of myelin antigens. Here is the myelin basic protein and a particular epitope of myelin basic protein, which is recognized more frequently in patients with multiple sclerosis as compared to age-matched control individuals. These have been mapped in great detail. And here's this T cell receptor again from a patient with multiple sclerosis, and able to precisely map how these epitopes of myelin proteins are recognized by the CD4 cells in terms of T cell receptor contact residues and residues that bind into the MHC molecule. But what's interesting is one can actually make a transgenic mouse with these T cell receptors. And here we took this T cell receptor from a patient with multiple sclerosis, recognizing this dominant epitope, myelin basic protein, and made a mouse which had this T cell receptor with the human MHC. And one can see by this graph that these mice spontaneously developed an inflammatory autoimmune disease that looked very similar to multiple sclerosis by having these T cell receptors from an MS patient. Interestingly, though the T cell receptors began by recognizing one epitope from this myelin protein, with time one can see that there's recognition of other parts of this myelin protein in addition to recognizing other proteins. This is a phenomena called epitope spreading. In other words, even if you start with a T cell receptor recognizing one peptide, with time, there'll be spreading of the immune response and other T cells will be generated recognizing other cell proteins in the myelin. Thus, efforts to do new therapies which target the T cell receptor or a particular myelin epitope are unlikely to work. There are newer techniques we've used to be able to indicate that MS indeed is an autoimmune disease, in that we can make tetramers of the MHC molecule loaded with self-peptides, which greatly increase the binding of the peptide MHC complex to the T cell receptor. Another major protein that's evolved is called Myelin oligodendrocyte glycoprotein, and we've used that to examine patients with multiple sclerosis. So we have here a flow histogram, which basically is a way of measuring binding. If one looks at the histogram to the right, one can see the frequency. Each dot represents a separate T cell, and one can see with a value of 5.68 this increased frequency of T cells binding to the MOG MHC complex. And we can show that indeed those are MOG reactive T cells by doing single cell cloning and showing that each of those little dots recognize, in fact, this self-myelin protein. But most importantly, if one compares patients with multiple sclerosis compared to control subjects, each symbol is a different healthy control. The open symbol, or patients with multiple sclerosis, in the closed symbol, one can see a half log to almost a log increase in the frequency of these autoreactive T cells. But note that normal individuals also have these autoreactive T cells in the circulation, which leads to the next fundamental point of what are defects in immune regulation? If we all have autoreactive T cells in our blood at some frequency, why doesn't everyone get multiple sclerosis? Well, that's because in order to get the disease, there need to be defects in immune regulation. And a very major population of cells called Tregs, which we'll discuss in a moment, was identified. And there's a great deal of plasticity of these Tregs. They can be driven to become inflammatory and no longer protective. So getting back to this brief review of immunology, we have the macrophage, which presents antigen to the T cell receptor. One can broadly divide the T cells into in Th1 types of cells, which are inflammatory and recognize intracellular pathogens. Th2 types of cells, which involve an allergy and asthma. They recognize predominantly parasites and turn off T cells. And two newly identified populations of cells, the Treg, which expresses a transcription factor called FOXP3, and the so-called Th17 cell, which is involved in inflammatory function to protect the gut and the skin. We now believe that multiple sclerosis is caused by both Th1 and Th17 cells recognizing self-myelin antigens. You don't have to worry, there's nothing between Th2 and Th17. The Th17 cell was named based on the secretion of the inflammatory IL-17 cytokine. The other critical player, which we now know in immune regulation, is called the regulatory T cell, or Treg. It expresses the FOXP3 gene, which upon its deletion leads to spontaneous autoimmune disease both in mice and in humans. Identified by high expression of the IL-2 receptor. Again, one of the emerging therapies for MS is targeting the IL-2 receptor, and I'll get to that briefly at the end. But we now know that the Tregs are central in regulating immune function. So if one looks at this slide from a review published last year in the JCI, we have this pathophysiology of multiple sclerosis, where antigen-presenting cells present self-antigen to T cells. Perhaps the antigens are cross-reactive with other microbial antigens, leading to activation of autoreactive T cells. In normal individuals, there are regulatory T cells and another type of regulatory T cell called the TR1 cell, which suppress these T cells from causing damage. Whereas in multiple sclerosis, there's lack of function of the Tregs and of the TR1 cells, allowing the activation of autoreactive T cells and autoimmune disease. This is very likely to be the fundamental pathophysiology of multiple sclerosis. There are likely to be other defects in different T cell populations, but we're beginning to develop a reasonably good working hypothesis now as to what causes the disease. And I encourage you to read these recent review articles to obtain more detail. A number of years ago, our laboratory was able to show defects in these regulatory T cells in patients with multiple sclerosis. The open symbols near the bottom of the graph represent patients with MS. The closed symbols are age-matched healthy controls. And one can see this defect in suppression of the inability to suppress proliferation entering the cell cycle of T cells in patients with multiple sclerosis. I mentioned that this defect is not specific to MS, but is seen in a number of different autoimmune diseases, including type 1 diabetes and rheumatoid arthritis. More recently, we're able to ascertain a potential mechanism for why this occurs. Basically, one can take these regulatory T cells from the blood, stimulate them very hard, and then look at their cytokine secretion. If one looks at the red circles, one can see that to the right, the patient with multiple sclerosis had an increased frequency of gamma interferon secreting regulatory T cells that was not seen in a healthy control. And here is a summary of these data. We have control MS patients on the left, looking at the frequency of gamma interferon secreting regulatory T cells. On the right, the frequency of regulatory T cells secreting interleukin-17. And you can see this major increase in the frequency of regulatory T cells. We know these regulatory T cells secreting gamma interferon just don't work. And that perhaps is one reason why one has loss of immune regulation. And one can prove the regulatory T cells by looking at the FOXP3 locus, which I mentioned before, and one can see it's demethylated at that locus, indicating that indeed they are regulatory T cells. Here's the evidence that if you block the gamma interferon, you can partially restore the suppressive function. So we have here control in patients with multiple sclerosis. We see the defect in suppression in the control patient compared to MS patients. Then if you add anti-gamma interferon, you partially, but not completely, restore this loss of suppressive function in patients with the disease. So in summary, regulatory human T cells isolated ex vivo from the blood have this plasticity where they can be driven to become inflammatory. In both multiple sclerosis and in type 1 diabetes, they're defective in vitro function because of the secretion of gamma interferon. There are multiple defects, however, in regulatory cells, and there are other regulatory T cells that secrete IL-10 that are defective. Experiments in humans, clinical trials are now in progress where IL-17 and gamma interferon and other cytokines are being blocked as a potentially new emerging therapies. By having these information about the pathophysiology of disease, arms us with the knowledge to understand the emerging therapies that are being developed now and in clinical trial. Next question is, what is the genetic architecture of the disease? MS is a complex genetic disease. The evidence of disease risk in the population is approximately 1 per 1,000 individuals of European ancestry. The SIB risk is about 2 to 4%, which is about the same as dizygotic twin risk. Monozygotic twins have about a 30% concordance rate. That if one monozygotic twin has a disease, about 30% of the others have the disease. Now that doesn't mean that MS is 30% genetic and 70% environmental, because as I mentioned, I'll show you in just a moment, there are over 100 allelic variants that all of us in the population carry that define disease risk. These are good genes, but if you have too much of a good thing, it leads to the disease risk. So it may well be that there are some individuals who have a full genetic load of these allelic risks, which could potentially have a 100% risk of developing MS, and those types of investigation are presently under investigation. Back in the 1970s, I earlier had mentioned the MHC protein, the major histocompatibility complex. The one flavor of the MHC, so-called DR2, or as we call the nomenclature, DR beta 1 1501, was identified over 40 years ago as a major genetic risk factor in multiple sclerosis. And in fact, knowing this is one strong indication that MS clusters with the other autoimmune diseases, one, and two, that this particular molecule as a receptor for glutamic acetate has potential therapeutic implications. I'll just show two summary slides on work in genetics. About over a decade ago, the International MS Genetic Consortium was formed in order to interrogate the genetic basis for the disease. There's a recent paper published in Nature where the group showed that the identification of about 50% of the genetic risk of developing the disease. As I mentioned earlier, these genetic studies indicated a primary role for cell-mediated immune mechanism in multiple sclerosis. One can use this ingenuity pathway analysis to begin to identify different targets. And knowledge of this target, in fact, can potentially lead to new therapeutic approaches. But the genetics indicates that multiple different cell types are involved, including the Th17 cells, the Tregs, Th1 cells, Th2 cells, and antigen-presenting cells. But all these different cell types are involved in the pathogenesis of MS. Moreover, you can begin to cluster the different genetic variants against different autoimmune diseases. For example, the IL-17 pathway identified by IL-23 receptor and the STAT3 appears to be involved in psoriasis, MS, and Crohn's disease. And recent evidence again indicates that anti-IL-17 monoclonal antibody has therapeutic efficacy in MS and in psoriasis. Finally, let me end with some what are the environmental triggers before I move to emerging therapies. We know that vitamin D and their vitamin D receptors identified by genome association scans, smoking, high fat, and in fact, perhaps sodium intake. I'll present these recent data related to sodium intake. The work is going to be published shortly. We examined a group of normal individuals to measure the frequency of Th17 cells in the blood and of Tregs in relationship to the genetic background and in relationship to the microbiome. We did DNA isolation and 16S ribosomal sequencing to look at species diversity. And what we found was that, in fact, eating at a fast food restaurant was associated with increased frequency of Th17 cells. So we went on to explore the role potentially of salt and fat in the diet in inducing the inflammatory cascade. And just to show that surprise was that if you add small amounts of salt to the culture, you had basically a logarithmic increase in secretion of these inflammatory cytokines. That is, you can measure it by secretion, relative message expression, but clearly small amounts of salt had major effects in inducing inflammation. You can measure this also directly in the T cells by adding small quantities of salt with this major increase in secretion and expression of IL-17 in the cells. Not just IL-17, but the pathologic cells in the disease appear to have other cytokines, including GM-CSF, gamma interferon, and IL-2, and all these appear to be induced by salt. We believe this relates to the fact that salt concentrations in the peripheral blood are at a relative level about 150, 145 millimoles, which is significantly less than the interstitial fluid. When you bring the concentration to that level, one begins to see this inflammatory cascade. And if you feed animals with a high salt diet, this is an animal model of MS, EAE, high salt versus control, the animals get significantly worse and in fact develop a quadriparesis as opposed to simply a weak hind tail. This is not due to increasing salt concentrations in the blood, but is a direct effect of the salt in the gut. If one looks at small intestines, one sees the high salt diet induces these inflammatory IL-17 secreting cells. So increased sodium concentration induces inflammatory cytokines. Mice fed with the high salt diet are more prone to severe EAE. and it depends upon the ancient stress stimuli related to MAP kinase inducing NFAT5 and SGK1. So finally, what about response to immunotherapy and emerging therapies? Dr. Riley will discuss a number of FDA-approved drugs, including the beta interferons, glutamate acetate, Tisabrate, which blocks T-cell traffic, Gilenya, which blocks T-cell traffic at a lymph node, and cyclophosphamide. Here's from a recent review written by Dr. Peltier and myself. We examined the various paradigms of how to use the present therapies, including natalizumab and fingolimod for treating MS. So in terms of emerging therapies, I'll discuss representative numbers of these: anti-CD52, dimethylfumarate, and in clinical trial, anti-CD20 and anti-CD25. And briefly presented on both of these molecules. What I'll do now is discuss Campath, anti-CD20. I believe dimethylfumarate is about to be approved, so it will be approved by the time this is viewed, and the anti-IL-2 receptors further behind in the development. Rituximab is anti-CD20. It's a protein expressed on B cells. The B cells are involved in MS probably as antigen-presenting cells. B-cell depletion affects antibody production, cytokine networks, and B-cell-mediated antigen presentation, activation, T-cells and macrophages. Because not only can macrophages present antigen, but B-cells can also, when they do, lead to a very different program in the T-cells. The Hermes Trial is a randomized, multi-center, double-blind, placebo-controlled phase 2 study in MS, went for 48 weeks with 2 arms, one with rituximab, the other with placebo infusions. And one can basically see the results in terms of number of new lesions. Patients receiving rituximab, the anti-CD20 therapy, had a significant decrease in the number of new lesions as compared to the control. The pharmacodynamics of CD20 have near depletion of the CD19 positive B cells. CD19 and CD20 are both expressed on B cells, so we have to use CD19 to identify them because the monoclonal will mask CD20. After 2 weeks, there's approximately a 95% reduction of the B cells, which returns to about 1/3 of baseline by week 48. So we mentioned why we use CD9 and why not CD20. The other drug I'll briefly discuss is Alemtuzumab. In a recent study published in a previous study published in New England Journal, it was used as compared to interferon beta in early MS. That was a randomized, rated-blinded phase 2 active comparative trial with 334 individuals relapsing remitting MS with disease duration of 36 months or greater, EDSS of 3. The antibody is a humanized anti-CD52 antibody discovered by Herman Waldman when he was at Cambridge, University of Cambridge in England. It recognizes T cells, B cells, NK cells, dendritic cells, and monocytes. Anti-CD52 cross-linking triggers T-cell activation, and CD52 can serve as a co-stimulatory molecule, induction of regulatory T-cells. It's currently FDA approved for B-cell lymphocytic leukemia. It leads to the rapid, almost complete depletion of CD52 cells in the circulation, though some cells are left in the lymph nodes. The monocytes return to baseline within approximately 3 months, and the B cells return to baseline by 3 months, and in fact can overshoot. The CD4 T cells take approximately 60 months medium to recover. The CD8 T cells recover at about half that time at 30 months. In the CAM S223 trial, there are 3 arms, one with alemtuzumab at 24 mg, the other at 12 mg for 5 days, and compared to Avonex. And these trials, in fact, have showed a reasonable efficacy of Campath. I should mention that one of the major issues with the anti-CD52 therapy is the induction of other autoimmune diseases. Again, as I discussed earlier, there's this autoimmunity perhaps could be viewed as one type of disease with different genetic variants, but half are shared among different individuals. Perhaps it's not surprising that a reconstitution of the immune system, which occurs after anti-CD52, might lead to different autoimmune diseases emerging. So in summary, what do we know about multiple sclerosis? There are multiple target myelin antigens, not just one, precluding this as a potential therapeutic approach. There are multiple defects in immune regulations, including Tregs and TR1 cells. There are multiple allelic variants, predominantly in immune genes. They're not bad genes. It's not a bad environment in terms of environmental triggers, but a bad interaction between genes and the environment, with multiple environmental factors causing the disease. And just the last slide of just a few points. The multitude of different immunosuppressive treatments increasingly necessitates, I believe, neurologists with expertise in immunotherapy. Right now we're up to 5, 6 different treatments, and as they continue to emerge, knowing which therapy to use is going to be critical. Secondly, immunosuppression works in patients with relapsing-remitting disease, which involves T-cell traffic from the periphery into the CNS, but does not work in patients with secondary progressive disease or primary progressive disease. It's not clear why the therapies not work in primary progressive disease, but perhaps one can look at these diseases where the CNS itself becomes inflamed, T-cell traffic is not important, and immunotherapy does not appear to work. And finally, based on what I showed from genetics and the biology, I believe the application of new biologic findings in patients with MS need to be applied to decide which therapeutic approach to use. And so combination of this new basic science or understanding of MS needs to be applied clinically to better choose which of these different drugs to use. So I'll stop there and thank you.